From Microbe TV, this is TWiEVO, This Week in Evolution, episode 56, recorded on May 19th, 2020. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels LD.
Hey there, Vincent. Good to be with you from LD Home Studios again.
How long do you think you're going to be home? You have any idea?
Good question. So we're just chatting a little bit, getting into work about once a week, back to LD Lab Studios, I guess you could say. And actually, the whole university has now moved into what they're calling sort of the orange zone from the red zone. So there is a just sort of dipping your toe in reopening, putting in a lot of, things in place. So wearing masks at our research labs, also setting calendars so that we're keeping the number of people, the density of people in any given research lab bay at a low level, one person at a time, working in shifts, actually, in some cases. How about there? Is it similar energy, or is it still pretty much shut down?
Well, this hospital's been pretty busy, so...
Of course.
I mean, this morning I came in and the parking lot's full. I had to actually valet park my car, which hadn't happened in years. So there are a lot of people here. The university, the medical school is not full up yet, but the plan is June 1st to start ramping up in phases. But so we're...
I see.
We're not, I don't know what orange means, but like my floor is pretty much deserted. So a lot of people are not yet in.
Similar with ours.
Slowly getting there.
Yeah. Same here. And I'm hoping that it does become a little more feasible to set up in the, in my office, just because I'm in the, as you know, LD Home Studios is in the attic here. And as temperatures are warming up, it's like a Finnish sauna. And so by the end of our episode, I'll have a science lather.
Yes.
Sort of figuratively and literally going here. Yeah. Anyway. So Vincent, it looks like we have a really, a growing, I would say, collection of really good listener feedback that I would love to jump into, including, a request to look at a recent, piece of work that came out. And so I think the game plan is to do that and then to really spend some time reading some of the letters and sort of reflecting on them.
All right, let's do that.
Count me in. So the first letter or request comes from Kayam, and pointing out this brand new preproof, and we'll talk about what that maybe is in a minute here. But it's a emerging piece on SARS-CoV-2 evolution, and wanting us to sort of run it up the flagpole here. So this is from the journal Infection, Genetics, and Evolution. And they call this, the stage that the work is being shared, a preproof. Have you come across this before?
Never heard of a preproof, no. Wait a minute. So proof is like when it's set into what the page is going to look like, right? I don't know. I've never heard of a preproof. Have you?
No. So I'm, familiar with a proof, which is when, generally as a article after it's done with peer review, and before the final version is published, the publisher will provide you with a proof. And usually it's this breathless thing, like get this back and proofread this and get it back in 24 hours or else. That's not necessarily true, but I guess a preproof, this is sort of, I don't know, this is the first one I've seen. I think it's somewhere in between a preprint, which we've talked about a lot, and we'll see evidence of another one, to compare to this one in some ways. And then a proof. So certainly there's been some more eyes on it. So the fact that the journal has picked this up means that there's an editor or group of scientists who've looked at this, and it might even be sort of in the middle of peer review in a sense. This is a little bit of speculation.
Yeah.
But then it does say, there's some language kind of attached to it, that it's not sort of the definitive or final version of the work. And so it's probably somewhere almost in the middle of the peer review process, it seems like.
Yeah, I mean, the preprints are not yet associated with a journal, right? Because you're submitting it. But this is clearly associated with infection genetics and evolution. So they are either going to review it, it's in review, or it's been reviewed and it's waiting to be set into proofs. So we don't know where it is.
Yep. And I guess, in some ways this reflects just how fast the field is moving, right? There's all of this hunger to try to get as much information out there. And we've fallen into some pitfalls because of that.
Yeah.
And so anyway, that may be more of a philosophical conversation. But so this pre-proof, it's from Francois Bellou's lab. His group is at the University College London. The first author is Lucy Van Dorp. And I just lifted from the Bellou Lab webpage a quick sentence describing what they do. It's research at the interfaces between genomics, epidemiology, evolution, and ecology. And, a lot of those overlapping fields are highly sought after to try to understand what's going on with SARS-CoV-2 today. And so I think that there is some good sort of background or a good view to be taken through those sort of overlapping lenses. And we'll kind of move a little bit quickly through this. But so basically the gist of the work, it's all computational analysis or phylogenetic analysis. It grabs a little over 7,500 of the SARS-CoV genomes that are now available. We've been discussing this on TWiEVO the last several episodes at that nextstrain.org or the GISAID, GISAID website that's sort of a clearinghouse for a lot of genomes that are coming in from around the world of SARS-2. And so, one of the things that we've talked about a little bit is, first of all, how do you pick your genomes, right? So there's now, probably somewhere north of 10,000 genomes available, but they're not all, it's not sort of one size fits all in terms of the quality. So some of them that are getting deposited, so there's certainly a cutoff for what might be accepted. However, even then, there can be a lot of differences in the quality of the genomes that sort of come through the pipe. And so this work, at the very least, tries to do a sort of a second filter step, which is to take genomes that have high coverage versus low coverage, in the hope that then, as they're analyzing the variation, that it's not just technical problems or sequencing errors or even annotation pipeline errors. And we've talked about that a little bit too, right? So there's even on this virological.org, there are common sort of hiccups in the pipeline for even knowing what the mutations are. And there's these sort of common ones that show up that aren't real. They look like they're different from the reference strain, but it's just a consequence of the analysis pipelines. And so obviously there's no evolutionary impact for something that you just are miscalling, right? And so you have to be a little bit careful about those things.
I was going to ask you, so these are typically like Illumina or similar sequencing runs, right?
That's right. So this is a so-called short read technology where you take little fragments and then you try to assemble them onto
So you'll have coverage sometimes a few hundredfold per base, right?
Exactly.
So how does the quasi-species fit into this? Because my understanding is, as a virus comes out of a person, you got a whole mix of genomes. So you do this a few hundredfold coverage. And I assume the sequence they deposit is an average of everything. And you're not really getting any quasi-species information, but it's there, right?
Yeah. And so probably want to be a little cautious that term quasi-species is sort of a hot button issue for some population geneticists.
Mutant cloud, right? Or something. Mutant swarm.
Yeah. But what you're describing is that there's when you collect the virus genomes from an individual, right, there will be some variation even in those.
Mm-hmm. Right.
And so exactly right. And so what you do is you take the consensus, right? So if you have, like you're saying, there's 200 overlapping reads at this one position.
Right.
And in 198 of them, it's the same, but then there's two that are different. You would just throw out those two different ones, and then you would just go with the sort of the majority rules.
So those two, one could be an error, or they could both be sequencing errors, right? Or they could be bona fide variants in the population. So you're getting rid of that information essentially by throwing it away, right?
Correct. Yeah. And you're getting rid of all kinds of information. So we've talked about, that could be most of the game, actually. So we've talked a little bit about recombination, right? And so the short read sequencing is good, really good and highly tuned for pulling out those point mutations, the letter changes in the RNA. not so good at being able to discern recombination, and therefore some of those interesting patterns might be lost altogether because you're basically mapping it onto a reference that doesn't have the that view of recombination available to it.
The reason I bring it up is because most people are not aware of quasi-species, all right, in RNA viruses. And so, they'll see a news article about a mutation, and I want them to understand that the virus that comes out of everyone has many, many different sequences. It's not just one. Mm-hmm. That is what we are doing to facilitate analysis. But, on a 30,000 base genome, there may be thousands of different sequences in that little 50 microliter sample that you get from a person.
Yep. Yeah, and especially as you start to use some of the long reads and notice that there are all of these, most likely, non-starter forms of just strange, sort of splicing together the beginning and the end of the genome with the whole middle missing, or some, all kinds of this variation. So there's, and as we've talked about a lot with mutations, the sort of standing idea is that most of these are going to be deleterious and lost. So even, in whatever variation comes out of that cough from an infected person, and it's not going to be, like you're saying, it's not going to be that reference level thing that we're measuring. A lot of that variation is probably just going to fall flat. It's not going to represent a viable virus particle. And we're kind of blind to that, right? And so so there's all of these sort of classes of mutations that we don't even sort of consider. And then for the most part, they probably aren't consequential. They'll just fall to the wayside. But you're exactly right, Vincent. It's really important to kind of have that bigger picture and to take into account these sort of potential blind spots in the analysis that we do. This work included. Okay, so then you have all of these genomes, 7,500 plus, and then you start to align them and start to look for differences or do phylogenetic analysis. And so the first thing they do is to try to date the time to the TMRCA, time to the most recent common ancestor analysis. And this is pretty standard for a lot of this. It's not perfect. You can't pinpoint this. And in fact, for SARS-CoV-2, there's been, I think, at least 5, maybe more groups that have used the kind of algorithms that are available out of the box. In this case, they use one called tree dater. So you basically have a tree, and you're trying to infer from the variation that you see today among those 7,500 back to the original event when this emerged, or the sort of ancestral virus when they were all identical before all of the mutations started to accrue after the spillover into humans. And then using a molecular clock estimation, then you can actually try to go back and put this on a calendar. And so what they say is from the genomes they picked from the thousands of ones sampled out there and deposited from around the world, that they have a range from October to early December of 2019. Mm-hmm. And so that's totally consistent with what other folks are saying, what some of the evidence was that we've heard about in news stories out of China for the timing of this. There's been some ripples, right? So we've heard, I think, some news stories about, oh, wait a minute, there was someone with symptoms in California back in January, or someone with symptoms in December. And then, in some cases, sort of going back and, after the fact diagnoses. But I think most people, I think the main sort of bulk of the evidence still very much puts it into late 2019 as a time of emergence. And that sort of matches from this independent view, which is which is kind of interesting, right? To take the genetic variation that you see today from the virus samples and try to work backwards as well. And so that's a pretty solid theme of evolutionary biology in general, right? So if you're trying to date when a dinosaur was walking around, you'll think about the carbon dating. And as people have put together phylogenies, not based on Jurassic Park dinosaur DNA, but based instead on morphological features, the types of bone shapes, et cetera. That's how you start to kind of try to triangulate these dates. But it's maybe a little more of an art than a science. It's hard to really precisely do this. Mm-hmm. Just all of the messiness of the mutational process and the limitation of the tools that we have. Okay, so then once they've kind of stepped through that, then, so it's more phylogenetic analysis. And in fact, they spend most of their time sort of considering two ideas. The first is, and I think they use the phrase here, that as they look at the thousands of genomes, everything is everywhere. So, and that refers to where the samples have been collected around the world. And so basically you begin to see some of this is the same variation showing up in different locations around the world. And this, probably follows directly from the fact that our species is pretty prolific in terms of our global travel and our interactions with each other. If anything, that's, one of the things that sort of is a hallmark of this most recent pandemic is that the globe is connected through interactions and travel, and we've really lowered the barriers of international travel in a lot of ways. And so in that sense, it's probably not too surprising that as sampling has happened, and we will talk a little bit about how random or non-random the sampling continues to be, even though we're up into the high thousands here. But anyway, you start to see similar mutations showing up at different places. And so these recurrent mutations are so-called homoplasies. This is potentially independent evolution. So it's basically the virus, just through random mutation, hits on the same letter change by chance. And in fact, there's all kinds of tools to detect this and data like it. So they use one called Homoplasy Finder. And this is based on the principle of parsimony. So you try for the simplest explanation when you reconstruct the patterns of mutation that arise. And so usually kind of a hypothetical case of a shared mutation showing up, the idea would be from an ancestor. So it's one mutational event, and then as in this case the viruses diverge or go to new hosts, infect new humans, because the ancestral one had that mutation, that one event is shared between the two that you sample down the road. What they're saying is that actually, no, this looks like it breaks parsimony, that you have two mutations, or you have the same mutation, but it arose twice. It was two mutational events to get there. And so they do that. Right? Exactly right. Yep. And so that's the notion of the convergent evolution, independent evolution. And that turns out to be, for this data set, they propose that is the case for, roughly 200 sites across the genome. And so the reason they put that forward as something that's, interesting to consider is that, so the idea is that from time to time, if it's kind of like lightning striking twice. And if it's a rare event, but if you start to see those patterns, maybe there's something to it. Maybe there's some signal, an evolutionary signal here. So if there's selective pressures that are, ending, so among the sort of sea of mutations, if there's common selective pressures that then favor that same variant to show up again and again, that could be a mark of sort of adaptive evolution. However, and this is a big however in this case, viruses mutate a lot, and so these could also very much be neutral changes. So they can rule out that these were not brought somewhere by travel, for example. Yeah, so that's an important point, and so that sort of that gets into the importance of thinking about the sampling and how random or non-random it is. And so, depending on where are these? How are the people traveling? How are the strains being? Who are the individuals that are being tested and then actually having their strains sequenced? You want to kind of consider all of the things that might break those patterns, and then it comes down to the algorithm and the phylogenetic analysis. And so you're breaking parsimony, but sometimes that's actually life isn't parsimonious. Sometimes I think we can all sort of attest to that we're not always following the most simple paths forward, and that's true of biology as well. That's true of virus mutations as they accrue as well. And so there could be certainly some false calls where something that looks like the simplest explanation, it's like, something that's looks more complicated actually has a more simple explanation. So there's some caveats there. And so then what they do is to try to, weigh the evidence a bit more, is sort of consider some of the other patterns that are showing up. So for example, are these mutations, so are they non-synonymous? Do they lead to an amino acid change in one of the proteins encoded by the virus? And so the answer is yes. I think for the 200 sites that they put forward, 80% of them are non-synonymous. And so then you might say, oh my goodness, that seems like a lot. But then you have to remember that the degenerate genetic code, actually just the way mutations work, that's almost 2 to 1 that you'd just out of the box from total random processes. So 66% would be non-synonymous just under a kind of coin flip scenario. So it's really hard here for this and many other reasons to distinguish neutral from adaptive changes. And in fact, I think it's worth it to kind of step back and just think about where this virus is in its sort of, or the course of the pandemic in a sense, right? So this emerged, and if we take their analysis, the analysis from now another half dozen labs or so, that's late 2019, and then, in this massive, in some cases catastrophic, uncontrolled experiment in our own species, seeing it transmit so rapidly around the world, this is probably mostly pretty boring evolution unfolding before our eyes, right? There's viruses mutate. And so, as we've discussed on TWiEVO already, massively consequential sort of emergence or spillover into humans. And now things might be given the ease that this thing transmits. And I think we'll talk about more of this in some of our letters from the listeners, that, it might just be sort of a dull patch for the next several months in terms of the types of variation and the consequences of them. However, of course, there's massive interest, and should be, in trying to discern whether there are adaptive changes that could either alter transmission, could alter virulence, or could alter, sort of the effectiveness of immune responses, or of course also vaccine responses. And so that's why this is kind of on the, front plate, or the, high on the radar screen for a lot of scientists today, obviously including us here.
So there are non-coding regions in this genome, and I wonder, I'm not sure they made this part of their analysis, but if you look at those specifically with the same idea that there may be common mutations appearing in different places, you should see it in non-coding regions as well, because they have functions, right?
Yeah.
And that, to me, that would provide more evidence that it's not travel related. Actually, that's not a good way to put it. I would like just to see that outside of the protein coding region, because if they didn't see that in a non-coding region, I would say the method is not right.
I know. And that's exactly what you look for, right? As you're trying to kind of get signal from noise in terms of things that might have sort of a functional impact.
Yeah.
Are there patterns? So, for example, you can ask, what are of those 80% of the non-synonymous changes, how many of them are sort of radical differences in the amino acid versus sort of conservative, right? Where you sort of keep the same charge. But then, you kind of hem and haw back and forth, because sometimes conservative changes can be really consequential from a lot of other work that's emerged, and as you're kind of hinting at, even both synonymous changes in coding regions, or changes outside of coding regions, right? If you change, for example, the expression of something by altering a promoter element, or the way a splice site or something, all of it, those can be sort of, really drastic changes that just from gazing at the sequences, which is what we're doing here and what we're seeing in a large part in this sort of early work on the evolution of SARS-CoV-2. It's hard to really pin it down. And that's, part of why we're here, is that we really are trying to weigh sort of what you can look at through evolutionary analysis with then how do you push it forward with functional analysis. And so that's why we've spent a lot of time worrying about plaque assays versus PCR, and what are the, so you can generate a hypothesis or two here. And in fact, they narrow into one change, a non-synonymous change in the spike gene, different than that variant that has kind of got a lot of attention in our last episode or two, and people have been talking about. It's a different, so they see one in spike, but it's different. And then they, kind of promote a couple of other mutations in the ORF1AB region in 3 different genes. And they even go and try to sort of connect that. Are there similar changes that you see from SARS-1 or from MERS in terms of the homoplasy? That is, the shared mutations independently showing up now, not only for the strain of SARS-CoV-2, but also for those earlier pandemics. And, they see some stuff that's sort of nearby, but there's no kind of smoking gun here, of, oh my goodness, this is happening again.
Mm-hmm.
I don't think you see that. And so then in the end, I think a really nice kind of summary paragraph that I'll just sort of paraphrase really quickly. So, as pathogens are capable of doing these pandemics, SARS-CoV-2, very likely zoonotic origin. So everything they're looking at in terms of the dating, the kinds of mutational patterns, in their phylogenetic analysis, sort of similar to, I think, the kind of the mainstream science view that's emerging from all of the evidence that we've seen so far. They go on to say that they don't think the virus may not be fine-tuned to its human host following the spillover. And then they point out, however, it's near impossible to predict future trajectories for the virulence and transmissibility of horizontal transmitted pathogens. And they reference back to a book that I think was published maybe about 30 years ago. And I'm old enough to remember books. Do you remember these things?
Yeah, I do. I have a bunch of them still, yeah.
Before the days of pre-proofs and preprints and everything else, yeah. So then they go on, it is possible that the population of SARS-CoV-2 will evolve into different lineages characterized by variable levels of virulence and transmissibility. However, despite the existing phylogenetic structure, it is important to stress there is no evidence for the evolution of distinct phenotypes in SARS-CoV-2 at this stage. Mm-hmm. Yeah, and I think that, that's what we've kind of been trying to say for the last couple months. Yeah, yep. And so I think this is a nice summary of where we are so far.
So a couple of comments.
Yeah.
This is a nice paragraph. First, they say, as most but not all pathogens are of zoonotic origin. I can't think of one that's not a zoonotic origin. Can you? I don't know why they have the but not all in there.
Yeah, good question.
I always tell people every human virus was originally a zoonotic origin because that's how I think it anyway. And then, do you know, Nels, any case, I guess the answer is no, but I'm just going to ask you, where we have well-documented proof for, lineages, evolution into different lineages with different virulence and transmissibility in a human virus, say. Animal viruses are easier to study, obviously.
Mm-hmm, mm-hmm. Yeah, the first thing that comes to mind, I guess, is the influenza, our history of influenza in the last 100 years or more, and how it has become seasonal, and then different strains arise through the reassortment of the so-called the H and the A strains, right, that re-emerge from animal sources, kind of year in and year out. Getting back to your first point, though, Vincent. So I do think, so that notion that everything is zoonotic, I guess, from an evolutionary standpoint, if we went back to sort of the emergence of Homo sapiens in our kind of primate ancestors, there could be endemic viruses that, as our lineage sort of evolved into our species of today, that kind of came along with us.
Yes. That's for sure. I totally agree with that.
Yeah.
But at some point before them, it was zoonotic for someone else, or pre-Homo sapiens, right?
Yeah. And there's certainly spillover in both directions. I'm forgetting the other, from human to other animals, the reverse zoonosis. There's a word for that, but yeah. And then in terms of
reverse zoonosis is one of them.
Yeah. Yep. Yeah, and then in terms of the notion of like how to think about the distinct phenotypes, or even this is the hot button issue of, are there multiple strains out there, which I think has been sort of an unfortunate, like, kind of thrown fire on, or gas on the fire, right, for worrying about massive increases of transmissibility or virulence. Mm-hmm. I think it really is true that we have some blind spots in how, because there are, from the longer view of evolution, there are certainly different strains that emerge, and at different times, and have very different features, including, so SARS-1 versus SARS-2. But yeah, that sort of intermediate zone, right, where you have something emerge for the first time, and then what does it become sort of down the road, in terms of thinking about, like, how do you trace sort of the evolution of one strain to eventually branch into? Yeah. And even that definitely,
it's hard for people, but, as you very, very well know, the introduction of myxomavirus into Australia to kill the rabbits is a great example of how both virus and host evolve within the space of a year to reduced virulence, right? And that was an experiment, and we could monitor it.
Yep.
We don't have similar data for people. That's the problem.
Yeah, no, agree. And then we end up, I would say, with some of these almost like uncontrolled experiments, right? Where, and now for the first time, I mean, we're tracking it in a level of detail and resolution. And so it's trying to find that calibration of how to sort of not read too much into it, but also take advantage of all of these new resources that are, I mean, it's really exciting to be able to obviously sort of grab 7,500 genomes and then do some phylogenetic analysis and try to get some idea of what's going on. But I would say
that's why this paper is nice, because they're measured. They say, look, we're doing this because we're looking for this, but we don't see any evidence of it. And that's
right.
the way it should be, instead of getting people all bent out of shape like the last paper we did about, an amino acid change that it results in increased transmissibility, right? I mean,
no, that's right. And so, but then I think you put your finger on an issue here, which is then, this doesn't get a lot of attention. It just sort of sits quietly. Yeah. And so, but I think it's a measured balance analysis. And so how do we start to put that forward, even though it's not as provocative? And I think that segues actually pretty nicely into our second piece, that we won't kind of dive in as deeply at all into the preprint in this case. But what happened in this instance is that Newsweek picked up on, and it's just exactly like you're saying, right? It's, this provocative headline or title, and then kind of running with it. And so so actually I'll do the reverse. I'll start with a news article that was written about this preprint, and then we'll maybe unpack it backwards to get to the preprint itself. And so the headline that came out in Newsweek, I think it was just a couple days ago, is "Scientists Shouldn't Rule Out Lab as Source of Coronavirus," new study says. And, then and then it goes on to, sort of, take something that sort of is science-y or sort of feels scientific, and then to say that, hold on, maybe we should really be entertaining some of these ideas more seriously about lab release or, a genetic engineered origin of the virus. But then as you start to work backwards and look at the preprint itself, what you find is that, it has sort of one of these kind of catnip-like headlines but it's not supported by any of the evidence that's laid out in the preprint. Yeah. And in fact, it's sort of an ad hoc comparison using, it looks like you see a non-synonymous change here and a synonymous change here, but it's actually, when you dig in a little bit deeper, what the preprint tries to do is take some of the variation that you see in SARS-2 and then immediately compare it in sort of a non-traditional way to SARS-1. And yet there's no sort of phylogenetic treatment here. It's not put into in any context that an evolutionary geneticist would be familiar with. So that's, the thing that happened in that first preprint, where the first thing you do is build trees, and then you sort of start to do some of the, you're starting to compare to some of the other work to see whether there's something going on here. Here it's just like, hey, we just thought we'd compare these two things and then say some provocative stuff that might, like kind of launch some grenades almost. And so you end up with this sort of half-baked comparison between SARS-1 and SARS-2, which tries to say, oh, well, there's a change or two here that looks like SARS-1 near the end of the pandemic. So maybe this is already, quote unquote, pre-adapted to humans. And then if it's already pre-adapted to humans, then the danger is you start to kind of step down the path to kind of this conspiracy theory, not based in scientific evidence, but just in sort of these loose sort of threads, that because this has like just a sort of glimpse of something that you might see in SARS-1, maybe there's more going on here. And then the Newsweek kind of picks it up and they speculate even more. And then, try to do a little bit of something measured. So then there's a quote in here, and I'll just read it really quickly. Scientists who looked at the study at Newsweek's request said that this analysis is unconventional and uses techniques that are unproven. They cautioned against drawing conclusions until more research can corroborate the analysis. And so, it's like, and we're kind of getting caught in this loop too, is how much time should we spend debunking this kind of stuff?
That's right.
Totally. Versus, and I think it's worth it for a conversation on TWiEVO and to kind of make these comparisons and point it out. But the hope is that we can start doing better because, we can't keep sort of just playing this tune, because pretty soon after that Newsweek, sort of, let's get some scientists to sort of give the quote unquote other side of the story. then it goes into the politics of this, right? What is the current administration doing in terms of maybe trying to fuel the flames of the idea that this is a lab release, right? We're kind of drifting well out of sort of the scientific evidence-based views here. And it's really, as we've seen on several occasions, maybe to put it mildly, just not helping to work through an ongoing global pandemic.
So the whole Newsweek article is based on one sentence from this preprint, right? Where it's a throwaway sentence. Yep. They want to know if there's something very close to SARS-CoV-2 because, as they said very early in the outbreak, it was very transmissible among humans. So it entered humans really good. And as they say, there's no evidence for increased transmissibility or virulence happening during this whole, 3 million people who are infected.
Yeah.
So it was ready to go. And they're saying their idea is there more in an animal somewhere that's ready to go and that we should be concerned about, right? Because if we happen to be able to dampen down this outbreak, is it going to just come back? And I think that's really the point. And they say basically, even a lab should be investigated just as a throwaway because maybe that virus was in a lab, even though there's zero evidence that anything related to this was in any lab, right? So to me, that's the key is that if you don't have any evidence that this something almost identical to SARS-CoV-2 was in a lab, then it's just an accusation. So get some evidence before, because you could say whatever you want, right?
Yeah. Yep. No. And then that's I think that's part of the trick here. So, and it's important too, the scientific method is to be open-minded and to generate hypotheses. And so it's not to sort of foreclose on any possibility, but it's how do we weigh the evidence we have and then move forward in an evidence-based way. And so, so you're right, Vincent, the point, the idea, which I think, just from a scientific perspective is, certainly it's a hypothesis, but how much weight it will or evidence might accrue behind it. So the idea that because the virus, spilled over once, that basically that we're not recognizing that there could be a second spillover or that it's sort of smoldering somehow. And so, fair enough and there could be something to that. But at the same time, it's sort of like, again, kind of calibrating, that first spillover is having some pretty big consequences on the world right now. Where obviously it's it's transmitting really well. It's changed the way we live. And to me, that's, if I'm calibrating it, like there's strong evidence that if I go to a grocery store with someone who just recently got infected and get in really close contact with them, I think that's where I'm putting sort of my energy. Whereas the probability of a second, an independent spillover sort of in the region, not zero, but in terms of sort of the acute thing that we're dealing with now, probably not at the top of the list. So certainly, if we go back to SARS-1 20 years ago, it's not at the bottom of the list either. So that these things are going to happen. It might, and we might be on somewhat of an accelerating calendar given the increase in contacts with things like bats and other wildlife. But so it's almost like it takes that sort of seed of an idea, but then Newsweek in this case really runs with it in a way that I think just goes off the rails.
Yeah, it does.
Pretty quick.
Well, the other one thing I want to point out about this paper is that they, as you said, they make a comparison between SARS and SARS-2. And they make this statement that, SARS had to evolve more extensively in humans because it wasn't ready to go. And they actually say in the abstract, after SARS-CoV had developed, in the late phase of the 2003 epidemic, after SARS-CoV had developed several advantageous adaptations for human transmission, there's zero evidence for that.
Yeah.
There's no evidence for that whatsoever.
Yeah.
You have all we know is that SARS-CoV diversified more extensively than so far has SARS-CoV-2, but there's no reason to think it's because the virus was getting better at transmitting among people. There's just no evidence for that. So I find that a big flaw. In this paper.
Yeah, no, I agree. And so then in the end, I think it's actually a really good exercise. So we'll obviously put up the links to both the pre-proof and the preprint, but to compare the two pieces. I'm actually thinking about doing this. I teach a critical thinking class for our first year molecular biology students. And this would be, one of the things that I think would be really useful. So to read both And then, if you'll forgive me to kind of tune your bullshit radar a little bit here. So,
right.
yeah, right. So, to compare the methods. So the first one, pretty tried and true methods that are standard to phylogenetic analysis. And written in a way that you could go back and have a shot at reproducing it yourself. That's sort of, for a scientific piece, that feels like that's sort of the ticket to entry to being science, right? That you would allow someone else to try to replicate your findings. You'd provide the information so that someone could do that. Now, it's notoriously bad. You read a methods section, you have to go trace back 4 papers that were published 10 years ago or something. But it should be there. There should be a thread so that reproducibility is possible. In a lot of these preprints, Vincent, we're just seeing this kind of, vague pipelines, and there's no way that you could hope to reproduce anything. I think it's worth weighing that in the second thing here. What are the conclusions of the piece, and are they supported by evidence? We were talking for that pre-proof, how that paragraph actually, it seems like, it kind of rings in harmony in your ears when you look at the evidence and you say, oh yeah, that's a measured conclusion. It's not necessarily an earth-shattering conclusion, but it kind of fits the evidence. Versus if the conclusions there's no, what you were just pointing out about the SARS-1 and the notion of this adaptation to higher transmissibility. Show us the evidence, or otherwise that claim becomes a loose thread that ends up getting potentially picked up.
Exactly.
Yeah. What are the authors' backgrounds? So for that first case, a pretty well-established lab. So, this could be maybe a whole nother podcast. In some of my colleagues, we've been having, I think, kind of, interesting discussions and debates about, as a science, like your expertise and what should you comment on or weigh in on, or should you just drop everything and start working on SARS-CoV-2? And should you quote unquote stay in your lane? There's some really good discussion to be had there. At the same time, if I'm weighing this and it's a lab that kind of does this for a living, it might not be SARS-CoV-2, but a computational lab that does and has a track record of doing this kind of analysis. That's helpful. I'm not going to go into a lot of details, but let's just say that the preprint that got picked up in Newsweek, that if we look at the backgrounds, it might be a different story in terms of, is there any virology? Is there any coronavirus? Is there any, what have the authors been up to, in the past? I have to be a little careful here, right? Because there's always, we don't want to get stuck in the catch-22. You have to do something for the first time, and science depends on us doing new and novel things that are probably a little outside our comfort zone. But in these cases, where it becomes a news piece in a national magazine, I am concerned about the backgrounds and that we at least weigh that in the discussion.
Absolutely.
Yeah.
Absolutely.
Okay.
I found an interesting article that I want to just place at the end here. And you should take a look at this, Nels, if you haven't already. It's on virological.org. Mm-hmm. It's a thread called Tackling Rumors of a Suspicious Origin of COVID. It was started back when it was still called nCoV 2019. Remember those days, Nels?
That's right. 50
Anyways,
years ago.
It's by Bill Gallagher. Yeah. Do you know Bill Gallagher at all? Does the name
I've heard the name. I don't think I've met him in person or anything like that. Yeah.
He goes through a very interesting phylogenetic analysis and very nice analysis. For example, the furin cleavage site, he gives evidence that it came by recombination from another virus, and he finds some traces of it in another isolate. So he writes here, about an hour from this writing, I can go on my porch and watch a flight of microbats rise from hollow trees in the wetland forest at the rear of my property to skim the headwaters of Gum Creek for their nightly meal of insects. It is the peril of being a virologist that there are times I wonder about their virome. But right now, the grocery store is a far more dangerous place than my forest. We would all instinctively like to blame that on someone somewhere. The truth is that every virus of human beings represents a past accidental emergence, an innocent wrong place, wrong time thing. This judgment is based on facts and molecular evidence, independently judged by different analyses in the hands of eminently qualified scientists. A number of us have never worked together over long careers. Some of us do not even know each other by reputation, yet we have all come to the same conclusion. In China, in Scotland, North Carolina, Louisiana, Texas, New Mexico, California, Australia, the backbone of the virus was derived from a common ancestor of bat RATG13 and SARS-CoV-2, most likely in Yunnan province, from which bat RATG13 was isolated. Small segments of sequence, hundreds of nucleotides long, in a genome of 30,000 nucleotides were derived from viruses ancestral to other viruses only recently isolated in Guangdong province. The only laboratory in which SARS-CoV-2 was concocted was a natural one in a bat cave in a process that took decades, an accident of nature waiting for human contact.
Yeah, wow. That's really good.
Is it great?
Yeah.
Thank you, Bill.
Almost has, I agree. Almost has a poetry to it in addition to
it does, really.
Yeah, in addition to the science. That's really nice.
And then which Nels, the naysayer, will say, well, how can you rule out that this was not in that lab? How could you do that? But we have no evidence that there is. So why don't we focus on what we have evidence for? That's always been my argument.
Yeah,
right?
No, of course.
And you say, in science we make hypotheses, and they're always based on evidence.
Mm-hmm. Yep. No, of course. And I think we'll go a little bit deeper into that. I think this is going to come up. I haven't had a chance to read through all of the mail that we'll tackle now, but I think this theme will come up again, and we can go a little bit deeper on some of these points. But yeah, why don't we dive into the mailbag here?
Sure.
And because we have quite a few really great responses here. So I'll read the first one. This is from Anthony. And Anthony writes, SARS-CoV-2 has achieved the virus version of the American dream. Ease of human-to-human transmission, requiring no intimate or even direct contact. A or pre-symptomatic carriers. And the hosts most likely to die are those with less or least likely to reproduce or to be less fit. The last feature for most of human history would have been inconsequential to populations, if not beneficial at that level. As a speculation, SARS-CoV-2 is such a perfect fit, I wonder if it is not truly a recent emerging disease. Analogous to HIV, might have spilled over to remote populations, perhaps with tigers as an intermediary long ago. I've not yet finished listening to TWiEVO 53. Maybe the answer is there already. And that's from Anthony. So, tigers is a new one, I would say. And I don't think you will find an answer in TWiEVO 53 quite to that question. But some really good points here, Anthony, and really appreciate you raising some of the kind of building off of our conversation already. And so, yeah, the notion of transmission, which has been a theme both on TWiV and on TWiEVO and the other This Week podcast that Vincent is running, and thinking about the transmission. So there's certainly viruses that transmit more prolifically than SARS-CoV-2. But this, and that, I think this will come up actually in a later letter, but thinking about sort of how natural selection might add or act on and above that has been something we've talked about and we'll talk about a bit more. And then, I think that the idea of the smoldering in the background and where it came from, that's still, so, just as we're talking about, the evidence is pointing to bat caves currently, and even to places in China based on the sampling that we've done and then the accompanying phylogenetics of that. And I think what Vincent just read from Bill Gallagher pretty nicely sort of crystallizes the state of the art and some of the thinking about this. But it's important to remember that this is definitely, I think, a ongoing pressure point of a lot of scientists, Chinese scientists in particular right now, who are doing more sampling to see if what is there that's out there that might start to fill in some of the clues about the very first events. And so I think, that notion of the wet market so I think it's important to remember that that's pretty unlikely that was the spillover event itself, that somehow someone was in the market, there was an animal in the market, and the virus made the jump there. Now, someone who was associated with the wet market, who was in and around the other regions, given a lot of the extensive travel that folks are doing, and some set of events either through, and again, potentially an intermediary species so there was the pangolin hypothesis, which kind of rose and fell. There's still some reasons to sample that in other species, especially I think based off some of the history of coronaviruses. So thinking about not only SARS-1 and the connection with civet cats, or MERS and the connection with camels, but even some of our common cold strains that go back many more years, and their emergence from other lineages of coronaviruses and potentially spilling over from cattle, from cows in some cases, the OC43 strain that's still among us today. And so, yeah, there's still this really blurry zone, and that's part of the reason I think some of the more kind of conspiracy, or I should say maybe politically charged ideas have sort of had a place to roost, is that there still are some really big questions about the details of that spillover. And honestly, without a time machine and sort of the limitations of our evolutionary tools and sort of related epidemiologic tools, it's hard to know how much closer, how much resolution we'll get on that question.
I think to answer Anthony, the time to most recent common ancestor puts it, as you said in that first paper, Nels, it puts it, the end of 2019. I think that's the spillover time into humans. But others have looked at the furin site and estimate it went into the precursor of this virus 40 years ago. So in that sense, right, this has been percolating for a while, but I do think it's a recent introduction in humans, very unlike HIV, which took many years to spread from Cameroon and the initial spillover into larger and larger populations. But there was a long period where we didn't recognize that immunodeficiency for sure, because it was buried in a sea of other bad health issues that we didn't notice. But I think it's different now. And we would have picked this up it was picked up in Wuhan immediately when there were 14 cases of unusual pneumonia, right?
That's right. Yep. Yeah. Yep. No, both good points.
All right, we have one from Tia. I've loved subbing your TWiEVO podcast for so many reasons, but I thought I should share one you might not hear often. I teach homeschooled high school students social science, anthropology, and honors biology. I give them bits of your podcasts and bring up episodes in lecture and our seminar-style discussions. What began as me trying to keep up with science news and fill in the many gaps in my own knowledge has really benefited my students. Besides content and skills, I try to keep a conversation going about science as a way of knowing and also a culture. Some of my students have a powerful attraction to science from the get-go, but others are drawn in when I point out connections to their loves, such as painting, dance, drama, or literature. Several students have become fascinated with viruses, for example, because they started by crafting from paper or air-dry clay trios of enveloped, helical, and polyhedral models. Art can suck them into our shared wonder of how the heck a bit of nucleic acid that does not even have a metabolism can hijack cellular machinery and undergo natural selection. Anyway, especially for the ones who probably won't go pre-med or become researchers, we talk a lot about other jobs related to science, from informed portrayals of Neanderthals to cover art on journals. I try to help my students visualize many possible futures for themselves. Yet for some, science can seem like a remote alien world, certainly not a place they might fit in. When you two chat about this lab and that, run through genealogies of who taught or worked with whom, contextualize preprints, joke about editors, discuss funding problems, and recall conferences, you are not just making enjoyable conversation on your podcast, you're painting a vivid picture for non-scientists like me and for my students of how modern science works. As insiders, you might not know how uplifting this is. Getting a glimpse of a world in which meritocracy is so valued is a beautiful antidote to a lot of negative stuff in the world today. When you mention names of every ethnicity and nationality, discuss the work of male and female colleagues with unquestioning equal respect, and otherwise just do your normal thing, it is a balm. Thanks. Nels did not write this. Tia from Alexandria, Virginia.
Wow.
I don't think you could have written that, Nels.
Well, no, I didn't write it, Tia, but you're welcome to write whatever you want for TWiEVO, and we'll read it anytime. No, that is really an uplifting letter, and I'm just thrilled that we're having that kind of impact, and our podcast is useful in this way. This is spectacular.
Just by chatting now, just being ourselves. I knew it would work. Or I didn't really know. I kind of had a sense it would be interesting to some people, but there you go.
What's the old saying? Even a broken clock is right twice a day. No, but seriously, Tia, this is so nice. And yeah, I mean, this is it. Like, so that the idea that science can extend into sort of art and that it goes beyond just being a researcher or a doctor. There's so much depth and richness there. And so the fact that this is useful in the clearly very thoughtful teaching that you're doing, I think that's really nice. This is one of those letters, Vincent, that, it comes along once in a rare time where you kind of remember why you do the thing you do.
Yes, absolutely. I mean, obviously, Tia is a great teacher because she understands the value of incorporating this into her, not in her curriculum, but how she tries to portray science to students with varying interests in the field, right?
Yeah.
It's perfect.
Huge. Yep. Agreed.
Absolutely perfect.
Yep. Keep it up, Tia, and we'll try to do the same, but we'll see what happens. Okay, next letter. So this is from Randy. This one's a little shorter and a little more technical. So a colleague at BioFire, this is a local biotech here that is involved in a lot of diagnostic testing, both for infectious diseases, but including SARS-CoV-2. And we mentioned this, I think, maybe 2 episodes ago. A colleague said that the test that was just granted through the emergency use authorization for SARS-CoV-2 takes 45 minutes, not 30 seconds. Too bad it isn't just 30 seconds, Randy. Yes, so thank you for clearing that up. I think I said, based on this sort of microfluidics PCR technology that they use, that it goes like in a snap of a finger. I meant to say 30 minutes in the heat of podcasting there, but even that would have been a little ambitious. 45 minutes. Still, on the scale of things, pretty quick if we can get that kind of turnaround. Obviously, doing the test is one thing. The logistics of getting the samples where they need to be through the pipeline and then the result reported, now we can add some more time to that. But
that's right.
But certainly those kind of quick turnarounds are pretty useful and going in a good direction if the test is reliable. We've heard even in the last week or two, I think, some mixed reviews on some of the rapid tests that have come out from places like Abbott Labs, and how many sort of false negatives or false positives might sort of accrue. So there's all kinds of complicated things to keep in mind as we talk about testing and other testing and tracing. There's it rolls off the tongue, but the actually doing it on many levels is really complicated.
You want to take the next one since that was short?
All right, fair enough. So the next one is from Joel, and he writes, "Hello, Vincent. My name is Joel Mark, and I'm a huge fan of both TWiEVO and TWiEVO. Thank you so much for bringing this high level of information to the public. I was just listening to the TWiEVO 55 podcast in which you were saying that there's no evidence that the virus escaped from the Wuhan laboratory. Instead, you are more comfortable with the idea that the Wuhan wet market was ground zero. I believe that so far there is no sufficient evidence to either the lab nor the wet market as being the origin of the infection, although there is good reason to investigate both the lab and the market. The wet market is a candidate for the origin of the infection because it was selling many different species of live wild animals, some of which could have had the SARS-CoV-2, and it could have jumped to humans. However, most reports indicate they did not sell the horseshoe bat nor the pangolin, which are the 2 likely host species for SARS-CoV-2. The lab is also a candidate for the origin of the infection because it was studying many different species of coronavirus derived from wild animals. It is possible that someone in the lab accidentally got infected by this novel coronavirus. Although labs are skilled in preventing such accidents, they are not perfect. I think the origin of this infection remains an open question, and that there has been no convincing evidence for either the market nor the lab. Do you think that there is good evidence for the wet market as the origin? Should we study the lab to see if this could have been the origin of the infection? Thanks for all of the good work. Joel from Santa Monica. P.S. There seems to be sufficient evidence that the SARS-CoV-2 is not genetically altered / engineered. All right, Vincent. So what do you say? Wet market versus lab
I don't
versus,
think the wet market is a candidate any longer. I think the initial idea was that many cases were there, but clearly the first case ever picked up in China was not associated with the wet market, and many of the original cases were not. And in that virological.org sort of thread that I was reading earlier with Bill Gallagher, he went through some of the results, and he said they've only found SARS-CoV-2 in the wet market. Nothing, a little bit back from it that would suggest that something was introduced there from an animal. So I think the people got infected elsewhere and went into the market, and that's why it looked like it was an epicenter, but it's not. The spillover happened somewhere else, and we don't have any evidence for where it happened yet. I'm sure they're working very hard to figure that out.
Yeah. And then maybe I would just weigh in a tiny bit on sort of the lab idea, so to kind of follow that rabbit hole a little bit. So, it is true that this, as the lab has been sampling and working with coronaviruses and other wildlife-derived samples, that they do have a modest collection of such viruses. I think maybe the important way of trying to sort of think about that is to just, how do we weigh that versus how many viruses in that modest sampling compared to the number of them that are circulating in wild populations of bats and other animals. And it's not even close. I mean, the number of virus variants out there in wild populations versus the ones that scientists have been able to collect through sampling, it's sort of, orders of magnitude less. And then the second maybe point here that I think is worth kind of putting into some perspective is what happens at a BSL-4 laboratory versus what happens in terms of the types of interactions people have outside of laboratories with no protective equipment at all. And this laboratory and others like it are incredibly safe in terms of the amount of thinking that goes into what can get in, what can get out, and how humans are interacting with these samples is pretty remarkable, actually, what it takes to do this. And I'm not trying to say that humans are perfect and that mistakes don't happen. They do. But sort of the layers of, checkdowns and backup plans. It's a little bit, maybe an analogy that's useful is thinking about flying on an airplane. We get on an airplane and a lot of us are pretty uncomfortable. We're pretty freaked out. We think it's going to crash and a lot of us are going to die. But then if we look at the numbers, we're much less safe driving a car, given that there's so many cars on the road. There are not all of this sort of engineering things in place, the professional pilots versus the amateur drivers, all of these things. There might be some echo in that analogy to what happens at a BSL-4 lab versus what happens to humans that are just out colliding with wildlife increasingly as humans continue to spread across the globe or move into new, previously wild places. So I think, maybe that's another way of coming at some of the reason why a lot of scientists are pretty skeptical that there is a lab release, in addition to the fact that there's no evidence for it. Just that if there is something in there, you can't rule anything out, but it's again sort of what are the, most plausible hypotheses to pursue. That might be one way of thinking about it. The other thing is certainly, this comes up a lot, right? Should there be investigations into the laboratory given sort of some of the loosely drawn together things? Maybe. I mean, I think my sense would be that anyone at that laboratory is probably pretty open to the idea that, let's take a look. They're not hiding anything. And yet this becomes complicated, right? Because now as politics starts to get folded into the conversation, and now we're in a whole different ballgame. I think it's certainly for me as a scientist, I'm pretty always a little bit shocked at how quickly things can escalate based not on sort of plausible scenarios, but based on sort of political expedient sort of routes. And that feels like that's what's happening in this case to me.
And what really bothers me is it becomes politicized and then people lie. So a few weeks ago, Mike Pompeo said he saw evidence from Five Eyes evidence that clearly implicated that lab. And there is no such evidence. So as a scientist, this bristles me because I want to see the data. And so don't tell lies just because you have a political agenda. Okay. Don't use science in that way. That really bugs the hell out of me. And this lab didn't have this virus as far as we know, because they never published it. And, when scientists get viruses, they publish them. And if you think they had this and they kept it secret and it happened to be the one that got out and caused the pandemic, then you're out of your mind. Just no way that's happening. And how many spillovers are there that never go anywhere? You think this one particular one that they happen to have was the one that it's just, we talk about risk on TWiEVO about risk of getting infected by doing different activities. Well, this is another example of how likely is that to be. Really down very low on the list, I would say. So,
yeah. Well, and maybe another way of putting it too is, so, there's a lot of politics lining up behind sort of really pointing out the regional origin or the Chinese origin. There's also politics on the other side in China trying to purport that there's a U.S. origin to the virus. Yes. And so then, we look at that and we're like, oh yeah, sure. Great one. But think about the perspective on the, of what the implication is of it emerging in China, right? It's the same sort of, this is a political expediency. And, unfortunately that can get some real traction. I think in the point you brought up about Pompeo, I saw a news piece after that, or a journalist asking him to pushing him a little bit farther on that. And he wouldn't even confirm that he said that. And yet I think the politics of it is to just sort of float it out there and then not really stand behind it.
Yeah, that's right.
And right. And so then you've kind of, it's completed a political task, but then what we all need to weigh is, how do we balance politics with science? And that's, again, this is probably for, more like cocktail hour than the end of our podcast. But, okay. One last letter, for today. We're running a little long here. So, did I read the last one? Do you want to take this one?
Sure, I'll take Ben.
Vincent?
Good afternoon. I'm a lab technician working for the Department of Defense in Washington, D.C., and your content has been keeping me sane. I will start out with my question. With all of the talk of possible antigenic drift and mutation, does the fact that the spike protein is the only readily available antigen on SARS-CoV-2 lessen the likelihood that there will be antigenic drift? The spike is extremely specific in its function in binding to ACE2 receptor, and any mutation in this is far more likely to be deleterious than beneficial, since the significant changes in protein conformation needed to prevent antibody binding would likely reduce affinity for the ACE2 receptor. Oh, why don't we tackle that first?
Yeah, it's such a good question.
What do you think, Nels?
Yeah, I think it's completely fascinating. And, and so one of the if we step back for a moment and actually think about other ongoing pandemics, right? So HIV or influenza and how difficult it is to mount either a natural antibody response, or to come up with a vaccine that sort of approximate ones or hits it there. So that's, that remains an open question, is sort of the trade-off between, as you put the selection of either of a natural antibody response in a host, or if we're starting to do vaccine-based interventions, is then in the presence of that pressure, what happens next? As the virus populations are sampling mutations, does it hit on one that avoids the recognition by the immune system? But is there such a big trade-off that now you compromise the ability to recognize the receptor, which you need to get into the next cell to sort of propagate the infection? So, and this is lots of evolutionary geneticists, evolutionary virologists are, exactly in this space. It's a fascinating question. As you start to bring in the structural biology, right, to see how these things like kind of fit together or don't fit together potentially as you make changes. There's some really interesting work I'd put forward on parvoviruses. Colin Parrish looking at transferrin receptor and how that becomes really species specific as you sort of march down the courses of infections over time. I think the jury's still out on this one in terms of how much sort of evolutionary wiggle room there is for spike evolution. We've talked, a little bit about some of the mutations, even on, in the pre-proof from earlier in this episode about some of the variation that's coming up, but whether there is what the functional implications are so far in terms of immune evasion, as it relates to receptor recognition. There's still little, if any, vanishingly little, I would say, evidence so far but this is a place that's going to become obviously as source of intense and ongoing interest in the evolution of the virus host recognition as it's sort of then there's immune interventions, both natural and then potentially vaccine-based.
The way I look at it, Nels, is, this is a coronavirus. And as far as we know, they don't do antigenic drift. At least the human ones don't, right? And so, of course, this one could be different. But I think in virology, you have to look at history. You can't forget it. Like in all aspects of life, can't forget it. You can't forget history because then you will repeat it. So in this case, I don't think it's going to do antigenic drift because the other coronas don't. And I think they all have similar origins, but we will see.
Yeah. And there is, even in the last few days, I think some encouraging early returns on both natural immune responses and then potentially naturally immune responses both to virus and to vaccine candidates. All this is super early days, so I don't want to get too far out on a limb on this yet. But no, I agree with you. This doesn't seem like a similar situation to influenza in terms of the drift. And so the specifics of that ACE2 and spike interaction, that's the right place to look. And I think you put your finger, Ben, on a really important question moving forward.
Very few viruses have the ability to undergo antigenic variation like influenza and HIV, right?
Yep.
Very few.
That's really true.
Or maybe not the ability, but do it. I mean, John Udell told us a couple of weeks ago, in the lab, you can select antibody-resistant variants of measles virus, right? You can do it with polio, but you don't see variation in the wild in people because for some reason, it doesn't have to happen. But it seems to be structurally feasible, at least to some extent. Does that make sense?
Yeah, I think so. And then, I think it's also important to remember just all of the, these, in some sense, we think of viruses as simple genetic elements, and yet there's so much complex biology here, right? So when the particles are getting packaged up, and so it's not just the spike, it's not just the glycoprotein or the surface, it's sort of how the whole thing deploys, the timing of it, the other, sort of structural and biological interactions that are happening between virus and new host cell, both on the surface and then as the replication cycle, it all kind of, plays together some way, or it gets read out by natural selection at some level. And so, my goodness, it gets complicated fast. And so, so actually it's a good point is, that might be, the sort of sloppy analogy of the front line. And yet there's so many other sort of battlefields that are all, sort of mixing together in a way that the outcome might not be clear. And so something that you could do, like the measles, there might be other biological features that's just never really becomes an issue because it's sort of constrained in other ways,
right.
That make it sort of the biology of it different. Yeah.
I put an exam question on my final exam in virology where they did transposon mutagenesis of the entire measles genome and the entire influenza virus genome, right?
Mm-hmm.
And you couldn't put much in the measles HA. Maybe it's an HN, I don't remember, the spike, but you could in the flu. So it's, again, analogous to what you said. It seems to be structurally constrained as to what you can put in it. So you can't change it as much. All right, continuing with Ben, I also wanted to make note of an issue I had with the way that Vincent has been arguing against those saying that SARS-CoV-2 is mutating to become more virulent or pathogenic. Please let me know if there is something here that is incorrect or that I am misunderstanding. I agree with Vincent's conclusion that there is no evidence thus far to support the claim that the virus is changing in any biologically or functionally significant way. However, when Vincent says, quote, it's doing pretty well, so there's no reason for it to change, end quote, he, or depending on who is reading this to you, seem to anthropomorphize the virus and begin to think teleologically about selection. Compared to others like measles virus or norovirus, SARS-CoV-2 is pretty bad at spreading between people. This means that there is plenty of room for increased virulence. I think you mean transmission, not virulence, which makes Vincent's argument a bit off target from an evolutionary viewpoint. To be clear, there is no evidence that the viable population of virus has undergone any non-neutral mutations. So the end conclusion Vincent is making is correct, but I think the way he argues towards it doesn't really hold up. As I said before, please correct me if there's anything here that is not right. Thank you for everything you do in improving scientific communication and in making the lives of those of us who don't have time for the full lit reviews every day much easier. So, if you know me, Ben, you probably don't. I am totally anti-anthropomorphizing viruses. I preach about it all the time. So I don't think you're right in saying that I'm doing that. I don't want you to think teleologically about it. There's no reason for it to change. It's not a good way to put it. I agree. But what the key is here, in my view, there is no selection. If you're going to argue that a mutation is emerging, there has to be a selection for it. And in my view, the virus is quite transmissible, so there's no selection pressure. So if I said there's no reason for it to change, that's not a good way to put it. I fully agree with you. What I try to say is that there's no selection pressure, and people who say, this change in the spike is emerging are not considering what the selection for that would be. I happen to think that SARS-CoV-2, you can't compare viruses when you talk about transmission, because a virus can only transmit in a range that is compatible with its family of other viruses. And SARS-CoV-2 is pretty damn good at transmission. It's not as good as measles, but it doesn't have to be. And so there's no selection for it to get better. That's my point. Norovirus has got a higher R0, although Noro actually is slightly higher than SARS-CoV-2, but not significantly. Measles is much more. But that's not the right comparison. Within the coronas, this is where they exist, and they do very well. And so that's why I think there's no selection for it. What do you think, Nels? How does that?
Yeah. So a couple of points I'd add to this. And so the first is just maybe circling back to the trickiness of not anthropomorphizing viruses. It becomes even more tricky when you try to not anthropomorphize the process of evolution, and then do that with viruses when you're trying to communicate sort of in the heat of podcasting. And so I've kind of let go. So, and that can, it also can be like fingernails on the chalkboard for evolutionary biologists to sort of the way we talk in shorthand to try to communicate. So it's a big challenge. And so anyway, minor point, but yeah, that, and it's absolutely, I think we're all on the same page with that. And then I would maybe just modify slightly the notion of no selections to be maybe, really sort of like just a sliver of like selection possible, which for all intents and purposes might be like no selection. But it's, I guess maybe I tried to kind of awkwardly address this last episode, which was to say, if you've turned the volume, you already have the volume at 10. To get the volume to 11, whether, 11 doesn't exist on most stereos unless you watch the old movie Spinal Tap, in which case he has the stereo that goes to 11. And so that's possible, right? That there could be a little bit more. But if you're down in the volume range of 1 and then you turn it to 2, you've just doubled the volume. And that's like natural selection. You can imagine that there's a lot of sort of wiggle room for natural selection to act at a lower transmission. Could it get better at transmitting? Perhaps. But then also I think Ben's the first question that came up, right, about the notion of drift and sort of the trade-offs in terms of receptor recognition. There can also, depending on the sort of complicated biology of the virus, there could be trade-offs above and beyond where transmission is now. And so in that sense, maybe there isn't an 11, but we don't know. I think what's happened is people have been acting as if the volume is at 1 and it's been turned to 11, whereas I think I share your view, Vincent, that we're already at 10. This virus is super good at transmitting. And so that it's not unlikely, and given history and other viruses in the family and others like it, that we're looking at most likely a sort of a boring patch of evolution for the pandemic. The real action happened, and we would love to see some of these events from the other questions. How did spillover happen, or what did the virus look like just before spillover? What were the key differences that happened to anything we've seen before that allows this thing to do so well? I mean, it's remarkable, right? I think I saw a T-shirt that had just the RNA letters, the 30,000 letters of SARS-CoV-2
Yeah, cool.
Printed. I'm tempted to get one, actually.
That's very cool.
But I would defy anyone to point at the letters that we haven't seen in that order before and tell us why this thing is so good at transmitting between humans. We just don't know. For all of the great, genome analysis, the 8,000, the 10,000 genomes that we can look at. You can go to nextstrain.org and spend all afternoon just sort of tinkering around. There's still so much we don't know about this. And so, anyway, but that's where we're working from, is to try to take all this evidence, all of the information that's coming from all kinds of different streams, and try to make some sense of it. And so, I think that's where we're standing. And so, I think Ben, Vincent, the three of us are on the same page here. And I just wanted to first thank you for your letter, and also thank you for your work as a technician at the Department of Defense. So glad that you're tuning in, and that we're helping keep you sane. So, we need you at the Department of Defense and others like you to be there to keep us sane, to have some more voices of reason, and to allow us to work together to have these conversations, to try to move forward in evidence-based ways. And so, I have to say, Vincent, all of these letters have just been great. And for me, a really fun way to sort of process some of this and to work through it.
Absolutely.
Yeah.
Let me just say, Ben, why you can't compare measles, norovirus, and SARS-CoV-2. They're different viruses. They have different structural properties, and they can only change within that, right? So you have to just look within coronaviruses and say, what do we know? And that's why you can't say it could be as good as measles, because it probably can't get that good, right?
Yeah. Yep. Very different, very different beast.
I think this is a great way to have a discussion. Now, I wrote a blog post a couple of weeks ago about there being just one strain of SARS-CoV-2, and people on Facebook, on the post itself, on Twitter, they're all many people are trashing me, saying, I don't know what I'm talking about, blah, 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 being nasty, which, I ignore because that's not the way to discuss an issue. I'm happy to discuss it calmly. If you want to write in a letter, Nels and I will talk about it. But,
definitely.
I think people don't want to listen, and they don't want to come to the end of TWiEVO and find out what we said. And they just they knock off stuff on Twitter, which takes them a minute, and they be nasty. And I'm sorry, that's not how you solve problems. I'm happy to get into a conversation about an issue that we disagree on. But if you're going to call me names, I'm just not going to engage, man. It's just not worth it.
I hear you. Yeah. No, this is so much more, nutritious, these kind of conversations and these kind of letters to really talk through it. And and it turned down that, it's really, that's the other thing is, the volume sometimes just, I've been talking with some friends just about how all the quieter voices out there, who are trying to work through this, aren't as in evidence. And it's easy to scream, just like you're saying. It takes a minute to write something kind of vitriolic on Twitter, but it's really these quieter conversations that come, now an hour plus into a podcast that it takes some real patience to dig in. Yeah. That's where all of, I think, the positive, productive stuff is happening. That keeps me sane. That keeps me optimistic, as well as to know that we're having this conversation on a deeper thread.
You got a pick, Nels?
Yeah, I do. So my pick of the week, if we were as we return to sort of evolution beyond SARS-CoV-2 at some date into the future, I wanted to lift up the current issue of the journal Current Biology, and it's that just dropped yesterday. It's a special issue called "The Cell in Evolution," and so I'll put up the link in our science picks. Really, I just have to say I'm like a kid in the candy store. So it's a series of essays and articles related to the emerging field of evolutionary cell biology. And so this is this idea that cell biology is compared to other major fields. So like developmental biology, we're all pretty familiar with the notion of evolutionary developmental biology, or EVO-DEVO, but hasn't been really as much attention to thinking about from a cell biological perspective, how evolution sort of unfolds in really interesting, complicated ways as cells go about their day-to-day business. And so already I have to say, just if you click through and look at the cover image for the issue, it's like, it just starts with a knockout punch. It's a electron micrograph of a photosynthetic, I think it's a single cell algae, Malamonis. And it's a single cell, but it's covered with these intricate scales and spines that are made out of silica glass, basically. And then these great protruding antennae. And it just, immediately shows you how elaborate evolution can get, even at the single cell level. And then as you dig in, and start kind of going through some of the topics. Some really interesting stuff. My old PhD advisor, maybe I shouldn't use the word old, my PhD advisor has an essay in there on the evolution of the ciliates and how they have 2 nuclei. And there could be an immunological reason for that at the single cell level, actually. How do you partition your genome or keep it protected from things like transposable elements or even sort of virus-like elements? And so, anyway, big recommend. If there's, I don't know what the situation is on open access for the issue. If you are looking through the contents and see anything that looks really, that you're curious about, and if you're unable to access it, please drop me a line either. So like my DMs, my direct messages on Twitter are open so I can accept, we don't have to be, whatever, friends or followers or something like that. Drop me a line and I will get you PDFs, or drop me an email. And, if you're not able to sort of get, access on it. But take a look. I think, to sort of step outside of some of the virus evolution that we've been necessarily focusing on, into sort of, it can just be almost soothing or therapeutic to think about all of the great evolution out there. And so that's my science pick of the week.
Nice. Very nice.
Yeah. How about you, Vincent? What's your pick?
Well, over at a company called Visual Science, who makes all kinds of molecular models of all things, but viruses in particular, they have made a new SARS-CoV-2 model.
Oh,
they
wow.
have model and animation, and so that's my pick. We have been working with them to make cover images for the next edition of the Principles of Virology textbook, which will be the 5th edition, and they made us 2 different viruses they made us HIV-1, which you can find on their website also, and SARS-CoV-2, which just came up. And so they're really pretty. So check them out.
Yeah, this looks
very
amazing.
Cool. Very cool stuff.
Yes. I'm just looking at the company website. Visual Science is a visualization, design, and communication studio that operates in a wide range, of high-tech fields, including science, biomedicine, blah, blah, blah. That's great. Kind of an echo of Tia's letter, right? The mixture of art and science, or visualize.
Sure is.
Really neat.
It sure is.
Wow. Very cool.
All right. That is TWiEVO 56. You can find the show notes at microbe.tv/TWiEVO. Send your questions and comments. You can send your questions and comments to TWiEVO at microbe.tv. If you like what we do, consider supporting us. Microbe.tv/contribute. My host here on TWiEVO is Nels Eldy. You can find him at cellvolution.org, on Twitter, Earlybird. Thanks, Nels.
Yeah, thank you, Vincent. Great discussion today. I can report that Aldi Home Studios is now officially in Finnish sauna territory. I broke a sweat. I'm sorry. Listening over here. No, it's good. I've got a good science lather going. Really enjoyed it. Good to be with you.
Now you can get outside, get some fresh air.
Exactly. Good idea.
I'm Vincent Racaniello. You can find me at virology.blog. Music on TWiEVO is by Trampled by Turtles. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, stay curious.